So back to lecture 21. So when we're looking at secretion, we have two types. We have a constitutive pathway, and these are proteins that are secreted at all times. So there doesn't seem to be a specific signal for it, although there may be modifications to the to the protein or sequences within the protein that direct the proteins towards this. So that's possible. Or it might just be a default pathway. And those are not necessarily mutually exclusive. At the bottom, we have an interesting form where we have signaling base. Okay, so in this particular example, we get a signal, and what that signal will do is when it binds, it causes release of the vesicular contents. Okay? So we had looked at the process of exocytosis, and we'll look at it in more depth today, talking a little bit about the energetics. But what we have here is a process by which a vesicle will fuse to, in this case, the plasma membrane for exocytosis. And what's important to note is that the membrane is actually intact throughout the process. And so we don't ever have a time where the membrane has a hole in it and allows things to flow in and out. A couple things to keep in mind here. So the cytosolic leaflet, so that part of the bilayer of the vesicle, fuses with the cytosolic leaflet of the plasma membrane, so that becomes one phase. And the extracytosolic, so that's the inside of the vesicle, will become part of the extracytosolic leaflet of the plasma membrane. And what this allows for release then of the contents directly into the extracellular space. And these contents, these proteins, if they're proteins, they haven't been exposed to the cytosol. So we're going to look at motor proteins today, endocytosis, phagocytosis. We'll talk about coat proteins, and then we'll look a little bit at membrane fusion. So we're going to start with motor proteins. Again, the way these work is that they move along these cytoskeletal elements. And these cytoskeletal elements are found throughout a cell. The two major ones that are used here are the microtubules. Microtubules more or less sort of span, they work their way sort of inside, spanning the cell. And then we have microfilaments, which are made of actin, and these are sort of located at the periphery or the edges of the cell, just under the plasma membrane. Motor proteins are unidirectional, so they'll move towards the plus or the minus end of the cytoskeletal element they're moving along. Okay, so all the cytoskeletal, well, the microfilaments and the microtubules have a plus and a minus end, so they're like one-way roads. And the process requires an input of energy. So I'm going to show you a video describing this in great detail. Oops. Okay, or...
Our brain is made of billions of nerve cells, and they're all connected. If we take a closer look, a nerve cell seems to have antennas. Most of them are receivers of information, but only one is a transmitter, called the axon. This axon is connected to several receivers of other cells, forming a gigantic neural network, the brain. Meet John. John is a kinesin, a motor protein. He lives inside a nerve cell, and he has a proper job. To ensure that a brain cell does his job properly, it needs the continuous flow of building materials, proteins. They travel through the cell using the cytoskeleton. If you would compare a nerve cell with a city, the cytoskeleton inside the cell would be the roads, and the traveling proteins would be the traffic. These materials are towed by motors along the roads. And just as in real life, there are different kinds of motors. and different kinds of roads. John's sole purpose in life is to deliver his cargo to a specific place in the axle. He takes the main roads, and he walks in just one direction only. John's job may seem easy, but it's not. He has to overcome a number of obstacles to ensure that the right amount of cargo arrives at the right place. To make the journey even more difficult, John is not alone. Other motor proteins ride along with his cargo. They haven't woken up yet, but that will happen soon. The journey starts in the center of the city, just like in the center of the cell. To enter the axon, John has to pass a place called the axon initial segment. In this segment, there are two kinds of roads. The main roads that John uses, called the microtubules, and a lot of little alleys called actin. And here, our brave motor protein meets his first challenge, because one of his sleeping travel companions, myosin, has woken up and starts to cling to the actin. And there are a lot of alleys. Only brute force can save John now. Fate strikes again. The other companion, Dynin, wakes up, and he can only walk in the opposite direction of John, resulting in a tug of war. But there can be only one. Along the axon in which John travels, there are places called synapses. Here, the axon connects to receivers of the other cells. Regulating proteins call the shots here. This traffic police makes sure that all passing traffic gets to the right destination. If John's cargo is needed in this synapse, he will be stopped and myosins take over his load. But today, John's cargo is safe. But what he does not know, that his road is under construction, just a few blocks away. In our nerve cells, the cytoskeleton is changing constantly. Roads are built, but are also broken down. Facing this kind of obstruction, John has to find a detour. John isn't the only motor protein on the road. There are many more. Our nerve cells need a smooth traffic flow in order to perform well. A traffic jam due to problems during the journey may ultimately result in brain disease. Understanding the challenges John faces could improve treatment or prevention. Finally, John arrives at his destination. He has fulfilled his destiny. But several other Johns are just getting started.
Okay, so a fun and surprisingly accurate depiction. So perfect. So we're going to see in the next section, we're going to be looking at nerve signaling. And that's one example where we have to deliver those neurotransmitters to the synapse. And of course, we know there's other types of exocytosis going on as well in the cell. Here we're looking at endocytosis, and if you look at this chart, it looks pretty much exactly the same as the exocytosis one. It's just going in the opposite direction. So with endocytosis, what happens is we're bringing materials into the cell. And it may bring them in specifically, so specific things it might pull in, or it might just bring in a random assortment of whatever happens to be sort of floating around at the exterior of the plasma membrane there. And so it starts by basically the membrane itself, the plasma membrane just invaginates, so it pulls in, and that either traps material that was just at the surface, or again, this might be specifically targeted via receptors. We then get a fusion of the membrane, and so again, as we saw with exocytosis, when we get the fusion here, what that does is it preserves the integrity of the membranes so that there's never a point where materials can just leak through, and then we get the pinching off of the vesicle. And so this will then move through the endomembrane system. So the vesicles then, they can move either towards the lysosomes or back to the Golgi, so they enter into the pathways of that first slide we looked at today. We'll spend a little bit more time, quite a bit more time on endocytosis in a few minutes. The other way that we can bring things in is through phagocytosis. This can bring much larger materials in. And if you look at the bottom, this is the arm of an amoeba, and it's pulling in an entire bacterium. So when you're looking at vesicles, they're about like 100 nanometers or so in diameter, so they can't really bring large materials in. But through phagocytosis, you can bring in things like entire bacteria. And the difference here is that it's not mediated by a pulling in of the membrane. What happens with phagocytosis is the cytoskeleton rearranges and pushes the membrane out to engulf whatever it is and bring it in. And some things, such as amoeba, they utilize this for feeding. Okay? So they can use this as a nutrient source. If we're talking about humans, does anyone know there's only one system that uses this for humans, phagocytosis? Do we know what it is? Yeah, the immune system uses this. And so we do this through like neutrophils and dendritic cells and macrophages. These can undergo phagocytosis. But the rest of our cells, under normal circumstances, don't. So we're going to look a little bit at receptor-mediated endocytosis. There's different types of endocytosis, and the one we're focusing on is called clathrin-mediated. And clathrin is a coat protein, which we'll get to in just a minute. So in this particular example, what we have is it's the coat proteins that mediate the invagination of the membrane, and they're associated either directly or indirectly with receptors. And those receptors bind to what's called a ligand, and that's a ligand is just a fancy way of saying something that binds. And for clathrin-mediated endocytosis, I think it's about 50 different receptors that are possible, recognizing 50 different substrates or ligands. So as the endocytotic process occurs, those proteins or ligands that are associated with the coat protein get brought in, okay? The vesicle then uncoats, and then those ligands can go through different pathways onto the lysosome, or maybe they'll be through retrograde transport brought back to the Golgi, or whatever it might be. But the important thing is here that we're bringing in specific components. And a well-understood receptor is the low-density lipoprotein receptor. So this is the one we know as bad cholesterol. And the receptor for this is associated indirectly with the clathrin coat. So the clathrin coat protein doesn't bind the receptor. There's an adapter protein that will allow it to bind. So a single, what we call a clathrin-coated pit, that's when we start to pull it in, might have up to 1,000 receptors, therefore bringing potentially up to 1,000 LDL particles into the cell. If we look at LDL, what this is it is composed of a lot of cholesterol and phospholipids. And in fact, it's a form of cholesterol called cholesterol, which is not actually mature. And this cholesterol gets activated in the lysosomes. So this goes through endocytosis, and it ends up in a lysosome, which activates the cholesterol particles. As the pH drops, again, that's what initiates the LDL from coming off of the LDL receptor. In terms of the good and bad cholesterol, people are often interested in that. So what the LDL, the low density, when this is circulating, the theory is that this is bad. What it does is this is generally moving things sort of away from the liver towards the cells. And if there's too much of it, what happens is that the cells where it would normally be going, they actually decrease the amount of receptor they have. So they're taking less up. So if there's more in the bloodstream, they're taking less LDL up. And so you start to get an accumulation of these in the blood. And that can lead to atherosclerosis, for example, or other conditions. In contrast, high-density lipoproteins, good cholesterol, does the opposite. It brings cholesterol back to the liver where it's taken up, and if there's an excess, what happens is the liver sends the lipids for excretion. So this particular example, what was happening was they were fated for the lysosomes, but that's not the only fate that we can have for these receptor-mediated complexes. They may go back to the Golgi. They may also undergo what's called transcytosis. So in transcytosis, what happens is we move things through a cell. And so if, for example, we were to look at the gut, the enterocytes in the gut, so they have this, oh, perfect, it drew it right for me. So we have this apical side, which is in the gut lumen. So here's all your food and fecal material. And then this basal side, which is access to the body. And what can happen is through the process of transcytosis, for example, something can move in to the cell and then out the other side. And that's a mediated process, and it's specific. So it's a way that we can pull specific things through a cell. And that's outlined down here in the bottom right corner. We can see a transcytosis. So this particular slide here is showing the different options for what happens when we bring things in an endosome. There's also molecules that can just get caught up as that endosome forms. And so this process is called pinocytosis, or cell sipping. It's a way that the cell kind of tastes the environment. It can bring in useful things this way, but it also is a method, again, of bringing the external environment in. Okay? And so that's called pinocytosis. There are other coproteins, such as caveolae. If things come in through the caveolae-mediated pathway, they don't go into the endolysosomal pathway. So we're interested in the clathrin-coated pits, the clathrin-mediated pathway, which joins up with the endolysosomal pathway. And that's the one we're focusing on. But just beware that there are other mechanisms for bringing things into a cell. If we look at the rates of endocytosis, for example, we can see here macrophages, which are immune cells, ingest 3% of their plasma membrane per minute. So this is a huge amount of membrane being brought in. So we know that there must be an equivalent amount of exocytosis occurring, right? Because we have to balance endo and exocytosis, or our cells are going to get very big, or their cells are going to get very small. So here we're just looking at 3 different coat proteins. The way they function is quite similar, all right? But what they do is they originate in a specific compartment, so a specific organelle, and they're used for specific types of transport. So for example, if we look at the COP2 coat, the COP2 coat forms on the ER, and that's going to be vesicles that are bound for the Golgi. COP1, in contrast, these are used for retrograde transport, bringing things backwards through the Golgi and maybe even back to the ER. We have been talking about clathrin, shown in green. This is important for clathrin-mediated endosomal intake, but also clathrin is utilized by the Golgi for vesicles that are going out towards the lysosomes. So different coat proteins are used under different circumstances. I mentioned that there's another one that's used externally called caveolae. So that would be another example of a coat protein, but it doesn't meet up into our endomembrane system, and so we're not really focusing on that one at this time. So the reason that these coat proteins are important is that a plasma membrane isn't going to just invaginate on its own. And so it's the coat proteins that actually drive that pulling in of the cell membrane. If we look at clathrin, it forms what's called a triskelion, which is this sort of three-legged structure. On the upper left, you can see a transmission, or actually that might be a scanning, I think, image, electron microscopy image of the protein itself, and it really looks like, I don't know, kind of three little legs. It's similar to the flag of the Isle of Man, if you're familiar with that, which is three legs in a triskelion. So this will spontaneously come together. It's favorable for them to come together. And we know that we can mix a favorable and an unfavorable reaction. So what we do is we mix that favorable formation of these clathrins coming together with the unfavorable pulling in of the membrane. Oh, and we have another video. It's just videos all day today. Brilliant.
Eukaryotic cells take in extracellular molecules through a process called endocytosis, in which the plasma membrane invaginates and pinches off cargo-filled vesicles. This movie shows a series of electron micrographs that have been artificially morphed to show the process of endocytosis as it may occur. The process involves a variety of molecules, including the cargo molecules that the cell takes in, the receptors that capture the cargo molecules, and molecules called adaptins that mediate contact between the receptors and the clathrin molecules that act to shape the vesicle forming at the plasma membrane. In the electron microscope, individual clathrin molecules can be seen as three-legged structures called triskelions. During the assembly of a clathrin coat, the individual triskelions come together, interact through their leg domains, and ultimately form a closed cage.
Yeah, and the natural process is also accompanied by creepy Halloween music. So those will actually form, if you have purified clathrin, these will actually form in the absence of the membrane. So you can form these in vitro. But what happens is through those adapter proteins, they're associated with the membrane, and so as they form, it pulls the membrane in as well. And so this is just a few different views of it. And in the top right, what what we're demonstrating is they're highlighting in green, yellow, and red 3 of the triskelions. So again, because this is energetically favorable, we can drive invagination of the membrane, which is unfavorable. So the site has to be selected where these form. And if we're looking at COP1 or we're looking at clathrin, it's a monomeric GTPase that determines where this forms, and that's called ARF. In contrast, if we're looking at COP2, it's a different GTPase called SAR that mediates it. And so what we start with is that the ARF will determine where this is going to form. All right. We start to form the clathrin coat. And so this is, again, favorable. We utilize a different GTPase to actually cut this off, to pinch it off. But then at the end, we have what's called this coated vesicle. So this vesicle is totally surrounded by that clathrin structure. And that's not much use, right? Because we want to be able to traffic that vesicle. We want to be able to send it to its destination. So what we have to do is we have to take those proteins off. So we have to strip those off. And that, of course, is unfavorable. So that requires an input of ATP. And now we have the naked vesicle, and you can see displayed on its surface here, we have some markers, and that will be used for directing the vesicle to whatever pathway it is going to go to. Here we have dynamin. Yes?
What was the other one that's ARF?
We have a slide coming up for it. So yeah, actually, I think a couple slides from now. So the other one is SAR. So dynamin, another monomeric GTPase. And what happens with this one is it's wrapped around the neck of the vesicle. And then what will happen is when the GTP in it hydrolyzes, it changes shape, so it actually just squeezes and physically pinches off the vesicle. We know that that's going to be unfavorable because when you're fusing membranes, some of those polar head groups have to go through the hydrophobic space. So it's always unfavorable to fuse a membrane. And so the energy from that comes from where? We have to input energy, which comes from the GTP hydrolysis. Okay? And so this slide on the right shows a membrane forming, and here is the kind of neck, and it's surrounded here by dynamin. Right? So just keep in mind for our monomeric GTPases, they're in 2 states, a GTP bound or on state, and a GDP or off state. They can be assisted in turning off via a GAP, GTPase activating protein. All right, and that speeds up the rate at which the GTP is hydrolyzed to GDP. To turn them back on, guanine nucleotide exchange factors are used. That kicks the GDP out and allows GTP to come in and bind. So let's have a look at how this could work. We're looking at ARF here. So this is a protein that in its GDP-bound state is floating around in the cytosol. This little tail, which is actually at the N-terminus, is pulled in, and this tail then, so this tail in this state is pulled in. When we exchange the GDP for GTP, there's a shift. The tail pops out, and the tail is myristylated, so it has a lipid on it, and this causes it to immediately embed into the membrane. And this, of course, is mediated by a GEF, right? And the GEF is located where the vesicle will form. So the GEF is membrane bound. Our friendly ARF floats by, gets exchanged, buries itself into the membrane, and that initiates clathrin coat formation. This is a picture from a different textbook showing how this works with COP2. COP2 is a little bit different in that the hydrophobic portion is actually part of the protein. It's not a lipid attached to the protein. And again, here the SAR1 GEF exchanges GDP for GTP, and in this case, this part that's shown in brown here is this amphipathic helix, meaning that one side of it, the helix, is polar and the other side is nonpolar, so it sits beautifully in a single leaflet of the membrane. You can imagine that. And so remember when we were looking at alpha helix, it's like a cylinder, and we can imagine that all the residues on one side of that cylinder are polar, and on the other side of that cylinder, they're nonpolar. So it just sits flat in the membrane with the polar exposed to the aqueous, the nonpolar to the intermembrane space. And so upon exchange for the GTP, it causes a structural shift that flops this amphipathic helix out, which then buries into the membrane and then initiates formation, in this case, of the COP2 coat. Any questions about that? All right. So that's our first kind of issue that we have, is budding off the membrane. But what about when we're looking at the opposite process for exocytosis? So how do we fuse a membrane? And so membrane fusion is mediated by a family of proteins called snares. So snares exist on both the vesicle, that's the V snare, as well as the target membrane, or the T snare. And so this is how our targeting works. And the targeting is mediated by a family of proteins called RABs. R-A-B. And lo and behold, they're also GTPases. So there's about 70 different RABs in the cell. And the RABs are important for getting a vesicle to the correct compartment. So the right place, and then it fuses at the target membrane. All right? We'll get into a little bit how we can recognize different membranes in our signaling section. But for now, I'll just say that membranes, even if it's within a single organelle, might have specific features on even a part of the membrane that are different from the rest. And so the cell can differentiate between different types of membranes. And this allows the vesicles, via RABs, to recognize different places in the cell, because it's not a random process. You send a vesicle out, it's going somewhere specific. It utilizes additional proteins called tethers, and these can stick a long way out from the target membrane and help to do this targeting. The guts of the whole system are that the T snares and V snares form a 4-helical bundle. 4-helix bundle, which is very stable and very energetically favorable. And that is what drives the unfavorable fusion of membranes. So let's look at this kind of the initial process. So we start by the RABs targeting. Here on the left and the right, we see these tethering complexes, which again might extend a significant distance out from the membrane to allow targeting. So the The tethers will bring the vesicle and the target membrane close enough that the snares now can interact, and the snares will spontaneously form this 4 helical bundle. And what that actually does is, as you're bundling those snares, you're making the distance between the vesicle and the membrane smaller, and you make it so small that it actually forces those membranes together. So it forces those polar head groups through the hydrophobic core. So you're literally using this mechanical energy of the formation of the helical bundles to drive, to squeeze those membranes together, which is pretty cool. We now have a problem, though, because we've now made a single membrane, right? So now they fuse into one membrane, and then we're going to have that 4 helical bundle, right, in that membrane, and that's super stable. Remember, it was so fun for it to form, energetically favorable. Now it's really stable, but we want to reuse those snares, so we have to input energy. And so ATP has to be put in at this step to recycle the snares. Questions about that? Yeah?
What does snare stand for?
Oh, it's like sensitive to N-ethylmaleimide soluble receptor protein or something. It's like, it's huge. I tried to memorize it before today's class, and then I just thought, no, I'm never going to get that right. No one's ever going to ask. So I'll bring the name to the next class. Here's a crystal structure of a snare complex. So crystallography is a way that we can determine protein structure. And you can see this beautiful 4 helical bundle. So we have 4 alpha helices, and they're bundled together. This one's a little bit weird. This is the one that nerve cells use, in that synaptobrevin is in the vesicle, and it's a single, it contributes 1 helix. Syntaxin, down here, contributes a helix. And then there's actually a 3rd protein called SNAP25, which has this region embedded in the membrane and provides the other 2 helices. If you look at this slide, you'll see a bunch of things that say BONTA, TENT. Those are sites where bacterial toxins cleave the snares. And so botulinum toxin, produced by Clostridia botulinum, it cleaves these snares and prevents release of neurotransmitter. Right? And what this does is this results in basically paralysis of the muscle. And so if you get even a little bit of this into you, you can die. If you put it in a specific place, however, you can cause paralysis of your face and get beautiful, smooth skin. And so this is what Botox is. Tetanus toxin also cleaves these on a different system, and that actually leads to the opposite of a tightening of the muscles that won't release. And so tetanus toxin, similar process, but the opposite effect. And again, that can also kill you. So we're going to be moving now to our next topic, which is neuronal signaling. And we're going to see here that, again, this fusion and release of the vesicles is important. And so, again, when we're looking at this is a neuron, if those vesicles can't fuse and release the neurotransmitter, it messes up the cell signaling. Again, in the case of botulism toxin, it causes paralysis and death. In the case of tetanus, it causes a lack of paralysis, so a tightening of the muscles and death. So we just want to point out the joy in this system. So 2 very potent toxins. And they inhibit the release of neurotransmitter, which we can see in this process here. Right? So let's just quickly, we have like 2 minutes, I'll just introduce what we're going to be looking at. So when we're talking about the nervous system, we have the neurons in the glial cells. The neurons are the ones that are kind of doing the work we associate with the nervous system. There's the nervous system we split into two, the central nervous system and peripheral nervous system. And the neurons, there's three different types. There's sensory neurons, which are, as they say, involved in like sensory perception. We have motor neurons, and these are for doing physical, or associated with physical activity, physical events. Sensory is with receiving signals. There's interneurons, which are involved in the processing of nerve signals. Glial cells play a support role, and these are what are called Schwann cells and oligodendrites, which I believe is in an upcoming slide. And so they're important for the efficient and for the functioning of the neural system, but they're not actually participants in it. This whole thing is going to come down to permeability of the membranes. And what's important is this section down here. Because we remember that cell membranes are impermeable to ions. So ions basically can't move through. They have to go through some sort of protein, either a channel or a transporter. And the other thing that's really important to keep in mind here is that the cell works very, very hard to maintain those specific ion concentrations on either side of the membrane. And we're going to see the importance of that in the next class. So I hope everyone has a good weekend, and we'll meet up next week.